Hello guys, welcome to Bosons Botany. So in today's lecture, I am starting with non-receptor tyrosine kinase or JAK STAT pathway. So if you have not checked out receptor tyrosine kinase, which I have already covered in my last video, then please go to that video before watching this. So I will link it up in the comment box or in the description box. So just visit to that video and then watch this one. Okay, so let's start. So rather than possessing intrinsic enzymatic activity, many receptors act by stimulating intracellular tyrosine kinases with which they are non-covalently associated. So in receptor tyrosine kinase, there was an intrinsic kinase activity in the cytosolic domain. But in NRTK or non-receptor tyrosine kinase, there is association of a kinase domain but non-covalently. That is, it is not permanently bound to the C terminal of the receptor. As you can see here in this figure, uh, there is one receptor which has been dimerized after the ligand, which is cytokine here. As you can see, after this ligand binds to the receptor, there is dimerization of the receptor, and this non-receptor tyrosine kinase, which is non-enzymatically bound to the receptor, is now close to the receptor and it binds to the receptor cytosolic part. Now, after the dimerization, there is cross phosphorylation by this non-receptor tyrosine kinase. As we all know, kinase means phosphorylation and removal of phosphate is done by phosphatase. So, since this is a kinase protein which is non-covalently bound to the C-terminal domain of the receptor, this protein will now cross phosphorylate each other, and also there is the phosphorylation of the receptor. So activation of non-receptor tyrosine kinase ligand binding induces receptor dimerization, leading to activation of associated non-receptor tyrosine kinases as a result of cross phosphorylation. The activated kinases then phosphorylate tyrosine residue of the receptor, creating phosphotyrosine binding site for downstream signaling molecules. Everything is similar to RTK. The only difference is that the kinases are non-covalently bound to the C-terminal domain of the receptor. And in case of RTK, that is receptor tyrosine kinase, this kinase activity was the intrinsic property of the receptor itself. And there is autophosphorylation, cross phosphorylation, then generation of phosphotyrosine binding site is also there, which was also similar in case of RTK. So there are few important points. These non-receptor tyrosine kinases include members of the cytokine receptor superfamily. So the main receptor which we can consider here is the cytokine receptor superfamily, and the ligand is definitely cytokine. So cytokine is needed for our immune system, and it eliminates the foreign particles from our body by using this signaling pathway. So the signal here is cytokine and the receptor is cytokine receptor superfamily. Like receptor tyrosine kinase, the cytokine receptors contain N-terminal extracellular ligand binding domains, signal or single transmembrane alpha helix, and C-terminal cytosolic domain. So the structures are quite similar to that of receptor tyrosine kinase. As you can see, there is the N-terminal towards the outside or the exoplasmic site. The plasma membrane will have the transmembrane domain having alpha secondary structure, that is protein conformation, which is having the alpha structure, which is the secondary protein structure, and the C terminal domain towards the cytosol. It is exactly similar like RTK or receptor tyrosine kinase. However, the cytosolic domains of the cytokine receptors are devoid of any known catalytic activity, which means there is no intrinsic kinase activity associated with the receptor. Intrinsic means it is not inbuilt property of the receptor. It is activated only when a kinase is associated with the receptor non-covalently. You know non-covalent interaction and covalent interaction. Covalent interaction will have permanent attachment to something or permanent binding to something, and non-covalent means there can be dissociation and association. So it is not a permanent attachment or no permanent bonding. So I hope this is clear. Now let's start with the signaling very quickly. Ligand induced receptor dimerization and cross phosphorylation of the associated non-receptor tyrosine kinase, which I have already discussed. After the ligand binds to the receptor, there will be dimerization of the receptor, then cross phosphorylation with the help of kinase domain, which is bound non-covalently to the receptor. Downstream signaling molecules that has SH2 domain. SH2 domain was also seen in case of RTK. So everything is similar here. Few differences which I am already highlighting, which I am already emphasizing. So you just keep them in mind. So HS2 domain was also in receptor tyrosine kinase. The kinases associated with cytokine receptors include member of genus kinase or JAK kinase family. And this is from where we get the name JAK STAT pathway. So the kinase, that is the tyrosine kinase domain, which has been formed after cross phosphorylation by the non-receptor tyrosine kinase, that domain or that site will be bound by JAK kinase, which is the first downstream protein or the first member to bind with the receptor after phosphorylation. The key target of the JAK kinase are the STAT protein, and this JAK kinase will further phosphorylate the receptor. As you can see, just this blue protein here, this blue structure of protein, which is known as JAK, this will bind to the phosphotyrosine site, and this will activate each other and also activate the receptor further downstream by adding phosphate to the receptor. And this newly added phosphate is now bound to the STAT protein, which is the further downstream signaling protein, which is activated by the JAK protein. So JAK will phosphorylate the receptor, and this phosphorylated part is bound by the STAT protein. And this binding is happening using the SH2 domain. As you can see, it is very clear in the diagram. Tyrosine phosphorylation promotes the dimerization of STAT proteins, which then translocate to the nucleus, where they stimulate transcription of the target gene. So the STAT proteins, after being phosphorylated or after being activated, will form dimer, and they will translocate into the nucleus. As you can see, it will translocate to the nucleus, forming a dimer, and in the nucleus there will be gene transcription of cytokine responsive sequence. So I hope this signaling is quite easy. So let me again brush up once again. The receptor is known as cytokine receptor because we are taking cytokine as the signaling molecule here. So before the signal is bound, the receptor is not dimerized. And after the receptor is bound to the signal, which is the cytokine here, there will be cross phosphorylation of the receptor, creating phosphotyrosine site, and this phosphorylation is done by non-tyrosine kinase protein, which is non-covalently bound to the receptor. After phosphotyrosine domain is generated on the receptor, first member to bind is JAK protein. This JAK protein will further phosphorylate each other, and it will also phosphorylate the receptor on the downstream part. The newly phosphorylated part will be bound by the STAT protein using SH2 domain. And now the activated STAT protein will form dimerized structure to translocate into the nucleus, and they will activate the cytokine responsive regulatory sequence, and the response will be generated. So this is a very important table regarding various signals and their receptors, showing this JAK STAT signaling or non-receptor tyrosine kinase signaling. So the most important signal that I have already discussed is the cytokine signal, and that cytokine will have further following proteins under cytokine. So for example, interferon gamma, interferon alpha, erythropoietin, prolactin. Growth hormone is also there in case of JAK STAT pathway. And growth hormone, don't get confused by uh, the growth hormone and the growth like epidermal growth factor or insulin growth factor, platelet derived growth factor, which are present in case of uh, G protein coupled receptor. So don't get confused by growth hormone and uh, growth promoting factors. So growth hormone is showing JAK STAT pathway or non-tyrosine pathway, but growth uh, uh, signal or the growth factor, not signal, growth factor like platelet derived growth factor and then insulin growth factor, they are present in case of G protein coupled receptor. So this is a very minute differentiation, and I have already dealt with G protein coupled receptor, so you can check that out. So this growth hormone is important because this is again a very crucial hormone for our metabolism. Prolactin is needed for milk ejection or milk production, basically after the birth of a baby, and erythropoietin is needed for blood clot formation, and interferon alpha and gamma, they are needed for showing the immunity or immune response, and the receptors are there like JAK1, JAK2, TYK2, JAK2. So these are some receptors, and STAT proteins are also mentioned, and their responses are also listed up here. So you need to remember the signals basically, and the responses. So I hope. So if you like this video, let me know in the comment section, and also I want to know what are the topics you wish to study, and just share my channel and subscribe to this channel for more such videos. And I'm trying to cover this series of cell signaling or cell communication. So I'm taking up major topics so that you can you know try for these exams, because these are some important topics which are covered up in such examinations. So all the best and keep learning. Thank you.